Hello, my name is Caroline Müller from the Department of Chemical Ecology of Bielefeld University. Here I present you our project ExpoSCREEN, effects of single and combined environmental stressors on the phenotypic plasticity of the ruderal plant Tanacetum vulgare, which we performed using the ExpoSCREEN platform at Helmholtz Center in Munich. Tanacetum vulgare is a fascinating plant. It exhibits different terpenoid composition in its leaves and flowers, which you can even smell. Different chemotypes can thus be distinguished. This plant species also responds plastically to stresses such as differences in water availability or herbivory. To set up the experiment, plants of different chemotypes were clonally propagated and sent from Bielefeld to Munich, where the gardener potted the plants and grew them until a proper size. Then plants were exposed in the ExpoSCREEN chambers, 4 parallel larger chambers, each consisting of 4 subchambers. The chambers have excellent light conditions and the outside climate can be exactly simulated. Each subchamber was assigned to a certain stress treatment. For drought stress application, plants were watered individually with more or less water. 2 weeks later, caterpillars were applied to plants of the herbivory stress treatments, either alone or in combination with drought stress. Before the first drought treatment, before the herbivory application, and at the end, so 5 days after herbivore feeding, plants were individually placed in a photostation. Here, each plant is automatically moved 10 times, and in each position a picture is taken. This allowed us to calculate the biomass of the background organs. Also, volatiles evaporating from the plants were continuously monitored using proton transfer reaction mass spectrometry. At the end, herbivores were removed and plants were harvested. Therefore, leaves were cut and shock frozen. 1 leaf per plant sample is now used for analysis of terpenoids via gas chromatography coupled with mass spectrometry. The other will be used for analysis of metabolic fingerprints using liquid chromatography. We expect to find differences in the total biomass of the plants depending on which stress treatment plant were exposed to. We also expect differences in their chemical profiles due to phenotypic plasticity. We aim to publish the results of this experiment in an open source journal. I would like to thank the teams from Helmholtz Center Munich and from Bielefeld University, and in particularly I thank the DPPN for funding this exciting project.